So good morning all of you. Welcome to the online botany classes. Today we'll study about genetic code. Let us start. So what is a genetic code? It is nothing but collection of base sequences found on mRNA, and these base sequences are called codons. So the codons correspond to each amino acids and to translation signals. So the codons that are found on mRNA, they actually correspond to the amino acids and to translation signals. What are those translation signals? That I will tell you while discussing in details about the genetic code. So as far as genetic code is concerned, it has been found that there are 64 possible codons that can be made out of 4 nucleotide base pairs. Okay, bases. 64 possible codons that can be made out of 4 nucleotides or 4 bases. You know, in case of RNA, you do get 4 bases. What are those? A, U, G, C. Thymine is absent. Instead of thymine, there is presence of uracil in case of RNA. So out of these 4, a codon consists of 3 bases. So if we consider that 3 bases can be found in a codon, and in total there are 4 bases, then 64 possible codons can be made out of those 4. Okay, 4 to the power 3 on the basis of 4 to the power 3. So genetic code indicates which codon specifies which amino acid. You can find out which codon specifies which amino acid through this genetic code. The concept of genetic code was first proposed by Gamow in 1954. Later, many workers have done a considerable amount of work on genetic code, such as Francis Crick, you know, the discoverer of DNA, Watson and Crick. So, Francis Crick in 1961 concluded that genetic code is triplet. What is triplet? Triplet means it is made up of 3 nitrogenous bases. Similarly, Nirenberg and Philip Leder in 1964 found out this 47 out of the 64 possible codons. So they found out 47 out of the 64 possible codons, and remaining 17 codons were discovered by Hargobind Khurana. Okay, it is known that you have to remember that the first codon deciphered was UUU, which codes for phenylalanine, and it was actually discovered by Nirenberg. Nirenberg found out that the UUU bases actually code for the amino acid phenylalanine. Got it? So let us now, so you came to know that what genetic code is. It is nothing but collection of base sequences on mRNA, and in total there are 64 possible codons. Okay, if we take into account 3 codons at a time. Got it? So let us now start the characters or the properties of genetic code. So the genetic codes have the following properties. Triplets. Genetic code is triplet in nature. It is COMMALESS. It is degenerate. The degeneracy character is found in case of genetic code. We will discuss about it in details in the following slides. It is universal in nature. It is non-overlapping. It has got initiation and termination codon. Then there is another concept of WOBBLE hypothesis, which gives a peculiar character to the genetic code. Then it is non-ambiguous in nature. Okay, so these are the 8 characters, 8 properties of genetic code that we will study. Triplets, COMMALESS, degenerate, then universal, non-overlapping, then it has got initiation and termination codon, then WOBBLE hypothesis, then non-ambiguous. So let us start the first one, that is genetic code is triplet. Okay, so the code consists of 3 nitrogenous bases on mRNA. Okay, in a specific sequence. It consists of 3 nitrogenous bases on mRNA in a specific sequence. So how it is written? If we take into account the 4 nitrogenous bases, that is A, U, then G, C. These are the 4 nitrogenous bases found in case of your mRNA. So how you will represent these 4 into the codon? You know that as it is written that codons are triplet in nature. That is, out of these 3 nitrogenous bases, 3 are present and these 3 represent a single codon. Got it? So it should be written as follows. It may be A, C, G or A, U, C, G, A, C in this way. So 3, 3, 3. So these are the, each one represents a codon. Each one represents a codon. Got it? So it is triplet in nature. Okay. Now come to the second property, that is code is commaless. So codons are read continuously without any punctuation. As I had written in the previous slide, it is written continuously without any punctuation. Got it? Then next one is code is degenerate. What is degenerate? Actually the concept of degeneracy was first proposed by Bornfield and Nirenberg. Okay. So what is this degeneracy? That is occurrence of more than one codon per amino acid. That means an amino acid is represented by more than one codon. An amino acid is represented by more than one codon. This is what the concept of degeneracy. So except methionine and tryptophan, these are 2 amino acids. All amino acids are specified by more than one codons. Okay, except methionine, and remember it, methionine and tryptophan, all amino acids are specified by more than one codons. So you know, you came to know what is degeneracy. Degeneracy means a single amino acid is represented by many codons. For example, your valine. Okay, valine is represented by GUC, GUU, GUA and GUG. So these are the 4 codons which represent the valine. So valine is represented by, let me write here, valine is an amino acid represented by 4 codons. What are those? GUU. Okay. Then another one is GUC. Another one is GUA. Then another one is GUG. So these are the 4 codons which make up the valine, which code valine. So valine can be represented by 4 codons GUU, GUC, GUA and GUG. Okay. Okay. Now come to the next one, that is universality. That is genetic code is same for all organisms. Okay, but there are some exceptions that I will let you know in the last slide. So universality means it is same for all organisms. Okay. Next one, that is non-overlapping nature. Non-overlapping means the consecutive codes, that is codes that are present consecutively, they do not share their codons. Okay, they are distinct, they do not share their codons. That means in this way, if the codons are CAT, 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 this is what your non-overlapping means. That means it's a separate codon, it's a separate codon, it's a separate codon. Non-overlapping. Got it? But had it been an overlapping one, how you would have read it? In this way. CAT, CAT, CAT. So it is the right one, that is non-overlapping. How it would, had it been an overlapping one, it would have been read as CAT, ATC, TCA. Okay. So this is not true, overlapping. But this is not true. This is wrong. Got it? So this is what your non-overlapping means. Okay. Then come to the next property, that is initiation and termination codon. So in case of your genetic code, there are presence of initiation and termination codons. These are translation signals, that is protein synthesis signals. The codon AUG that codes for your methionine, okay, methionine is an amino acid. AUG that codes for the amino acid methionine is the initiation codon in case of prokaryotes. Initiation for translation, initiation for protein synthesis, okay, in case of prokaryotes. Okay. Sometimes GUG also serves as initiation codon, but remember that these initiation codon or start codons, they are present towards the 5' end of the mRNA or 5' end of the mRNA. Remember it, the initiation codons are found towards the 5' end of mRNA. Okay. Similarly, there are 3 other codons, UAA, UAG and UGA. These are called as stop codons or nonsense codons. They do not specify, do not represent any amino acid. Rather, they specify the polypeptide chain termination. That means these UAA, UAG and UGA, they bring about the termination of protein synthesis. Okay. That I will teach you in protein synthesis in a separate video lecture for protein synthesis. But remember, these 3 UAA, UAG and UGA, these are called stop codons or nonsense codons, which specify polypeptide chain termination. They do not code any amino acid. Okay. And remember that these stop codons are found towards the 3' end of mRNA. Okay. 3' end of mRNA. Now come to next property, that is your WOBBLE hypothesis. What is this WOBBLE hypothesis? WOBBLE hypothesis says that according to Crick, your base pairing between bases in anticodon of tRNA and codon of mRNA, that you will completely know about how anticodon of tRNA and codon of mRNA works in protein synthesis, when I will discuss it in a separate video on protein synthesis. But remember that the anticodon sequences found on tRNA and the codon which we have studied on mRNA, there occurs base pairing between these two. Okay. So this base pairing follows strict base pairing rule for the first 2 bases. Okay. And for the third base, you know that codon are triplet in nature, that means made up of 3 bases. For the third base, it is not that stringent, not that strict. So Crick proposed the term WOBBLE at this site. I will tell you how it happens. Okay. So according to this, the base pairing between anticodon of tRNA and codon of mRNA are as follows. How? If the base in anticodon is A, then the base in your base in codon is U. Okay. But if the base in anticodon is U, the base in codon is A or G. Got it? Similarly, base in anticodon is Z, then base in codon is U or C. If the base in anticodon is C, base in codon is G. Okay. Similarly, another one that you might not have come across, that is inosine. If the base in anticodon is inosine, then base in codon should be A or U or C. So what is inosine? Now the inosine is produced by post-transcriptional enzymatic changes. Okay. Inosine is produced by post-translational enzymatic changes. Okay. Now, after WOBBLE hypothesis, another property of genetic code is that it is non-ambiguous. What is non-ambiguous? It means a particular codon always codes for same amino acid. So codon makes the same amino acid irrespective of their position on mRNA. Okay. So here non-ambiguous means a codon always codes for same amino acid. So the codon makes the same amino acid irrespective of their position on mRNA. For example, AUG, okay, it codes for methionine. We have already discussed about AUG. It's the initiation codon of protein synthesis. So AUG codes for methionine. If it is the fast codon, it will make the same amino acid methionine, even if it is second, third, fourth codon on mRNA. Okay. AUG codes for methionine if it is in the fast place, a fast codon. Even if it is present in the second, third, fourth position or fourth codon on mRNA, still then it will produce the same amino acid, that is methionine. So this is what the non-ambiguous nature of genetic code. So particular codon always codes for same amino acid. Okay. Now there are some exceptions to your genetic code. What are those? First one is that exception to universality. We have already studied that the genetic code is universal in nature, that is the code that specifies for a single amino acid, the same code specifies same amino acid is specified by the same code in each and every organism. So there are some exceptions like AUA. It codes for isoleucine. Isoleucine is an amino acid. It codes for isoleucine in mitochondrial DNA. Okay. While it codes for methionine in human nucleus. It should code for isoleucine in each and every case, but in case of human nucleus, the AUA, same AUA codes for methionine. Okay. So it is an exception to the universality character of genetic code. Another one, UGA. It is a termination codon in nuclear DNA. Okay. In case of nuclear DNA, it's a termination codon. While it codes for tryptophan in human mitochondria. Okay. It codes for tryptophan in human mitochondria. Okay. Now another exceptions to genetic code is ambiguity. You know, we have already studied that it is ambiguous in nature, that is the codon specifies the same amino acid irrespective of its position on mRNA. If it is present on the first position, it will code this same amino acid when it is, even if it is present in the second, third or fourth position. But there are some exceptions to it, like GUG. Now GUG codes for methionine if it is the fast codon from 5' end. Okay. If it is the fast codon from 5' end, the GUG codes for methionine, but it codes for valine if it is present in any other position. Okay. Ideally it should be coding for methionine in each and every position, but GUG codes for methionine only when it is present in the first position from the 5' end. But if it is present in any other position other than 5' end, okay, other than first position, if it is present in any other position, it will code for valine. So this is another exception to the genetic code. Okay. Now let us see the genetic code here. As you can see here, I have given you the list of various codes and the amino acids coded by these codons. Okay. And there are 3 codes which indicate the stop signal, UAA, UAG and UGA, and there is also initiation codon, AUG, methionine. Okay. So this is what your genetic code is. I hope the particular video would be helpful in understanding the genetic code. Thank you very much.